Hey everyone, and thanks for joining me today for episode 789 of People Behind the Science. I'm your host, Dr. Marie McNeely, and today I'll be joined by our guest, Dr. Karine Gaelle Le Roch, to talk about life and science. I hope you enjoy getting a behind-the-scenes look into the life and experiences of another researcher in today's episode. And if you want to meet even more of today's scientists, you can check out our website at peoplebehindthescience.com or subscribe to our show on your favorite podcast platform. And today, Karin is going to share some of her own insights and stories. So get ready to meet another one of our wonderful people behind the science.
Every day, discoveries are made that will change our understanding of the world around us. Dr. Marie McNeely is here to bring you the brilliant minds who are making these discoveries so they can share their incredible stories and take you on an amazing journey. Welcome to People Behind the Science.
Hello, everyone, and welcome to People Behind the Science. Today, I am thrilled to be speaking with our guest scientist, Dr. Karine Gaelle Le Roch. Karin, thank you so much for joining us on the show today. How are you?
Thank you. I'm very good.
Well, thank you so much for joining us, and I'm looking forward to learning more about you and your work in our conversation today, but perhaps we'll start with a little bit of background. Listeners, Karin is a professor in the Department of Molecular, Cell, and Systems Biology and director of the Center for Infectious Disease and Vector Research at the University of California, Riverside. She received her bachelor's degree in Biochemistry and Microbiology from the University of Paris-Sorbonne, her master's degree in host-parasite interactions in a joint program between Oxford University and the Pasteur Institute at Lille. And she received her PhD in Parasitology and host-parasite interactions from the University of Paris-Sorbonne. Afterwards, Karin conducted postdoctoral research at the Scripps Research Institute in La Jolla, and subsequently she was an Institute Fellow at the Genomics Institute of the Novartis Research Foundation. Karin joined the faculty at UCR in 2006, and she has earned various awards and honors throughout her career, including the Academic Senate Regent Award for Outstanding Research from the University of California, and she was a participant in the Provost Faculty Leadership Program at UCR and the University of California's Women's Initiative for Professional Development. And today, Karin, we're excited to get to know you as a scientist, of course, but also get to know you more as a person. So can you start by telling us, what do you like to do when you're not doing science?
So I first would like to thank you for the invitation. I'm really happy to talk to you right now. So what do I do when I don't do science? I take care of my son and I go to see him playing soccer, but I also love sailing and skiing. So if I have a bit of time available for myself, I go sailing and race in the San Diego Bay, or in wintertime, I also enjoy skiing a lot.
That sounds wonderful.
I actually enjoy outside activity and getting a bit of fresh air to help me think about my work. It's often very important to be able to disconnect, to think better.
Absolutely. I think getting away from the computer screens and the fluorescent lights can go a long way to clearing your head. Well, I'd love to talk about your scientific side as well. So how do you describe your work and your research to someone who's not in science or perhaps just not in your particular field?
So I've been trained as a scientist, but I only became excited about the work I was doing when I started working as an intern in a lab in England, working on parasites. I started working on parasites infecting young kids in Africa and worms transmitted by snail. This parasite was called Schistosoma mansonii. And I become really passionate by this life cycle of this very complex parasite that can infect a human around the world. And then I've always been really excited to actually focus on this parasite that usually infect people in developing countries that don't have easy access to health and medicine. And working for this population is definitely really exciting and rewarding for me. So today I worked mostly on the human malaria parasites that's infecting over 200 million people annually and can kill up to 600,000 people, mostly kids under 5. And my main work is really to try to better understand the parasite life cycle, how all of these molecular components inside the parasite work together, and how we can disrupt the parasite inside the human body and find new medicine to cure kids infected by this parasite and that lead to the disease.
Well, Karine, I think this is such an important area of research, and I'm glad to hear that you've got this passion. And I think it can be inherently motivating when you're working on one of these problems that is impacting real people around the world in large numbers. But I love talking about motivation because I think there are challenges in science, and sometimes you need a little quote or a saying or a mantra to just keep you going when times get tough. So do you have a favorite motivational quote or something that just really motivates you?
In general, when people say no, I have a certain tendency to actually love going against these people saying, no, you cannot do it, and love to demonstrate that, yes, you can. If you're highly motivated and consistent and hardworking, you can actually do what you've always been dreaming doing. So persistence and hard work is my way of motivating me, in fact.
I love that. I think it can go a long way in science. You really have to be persistent and motivated, and it can take time to learn this process as a scientist. So when you look back over the course of your career, are there particular mentors or role models or people who really helped you learn the ropes and get to where you are today?
I think it's very important to make sure you meet the right people and to make sure these people are giving you confidence. So when I actually decided, because I'm French, I was born in France, but I was okay at university, but not really passionate by what I was working on, and decided to learn how to speak English. If I wanted to be a scientist, I had to learn how to speak English, and decided to move to the UK and North Wales to learn science, to learn the work at the bench, but also to learn English. And I ended up in a lab with a really great scientist that was extremely supportive, and for the first time in my whole career, starting to talk to me as a real person. I have learning disability. I'm dyslexic, so it was always very challenging. Always had to work much harder than most of the people around me. But having someone that looked at me for the first time as a real person and did believe in me and was always giving me some encouragement and positive feedback was actually really important for me. And also, of course, became passionate about this parasite and the fact that I was working for kids in developing country. But yeah, you always receive a lot of negative feedback, deception, rejection. And very often, especially as women in science, people keep telling you that you cannot do it, that you cannot achieve it. And meeting some of these nice and supportive people around your way can make a huge difference.
Oh, absolutely. And I think meeting these mentors can completely change your career trajectory in some cases. And you mentioned that you perhaps had a change of heart that shifted you towards this area of parasitology. But could you rewind back even farther, Karine, and tell us what first got you interested in science at all?
It's mostly because I was dyslexic. I couldn't write anything. It was very challenging, but I liked molecules. And I always thought, okay, if you have a hard time writing, maybe if you become a scientist and deal with molecules and atoms and carbons, then it's going to be easier for you to actually demonstrate to the people that you're not that stupid. So it started like that, by trying to avoid as long as possible to write something. And the funny part is now, because I have to write grants, I spend all my life and all my time to write grants and manuscripts and publications.
Right.
But initially, I just wanted to avoid writing and really focus on small molecules.
Very interesting. And I think this can be challenging for people out there who are our listeners who might have a disability themselves, whether it's dyslexia or something else where they feel like science isn't accessible to them as a career path. Do you have any advice or things that helped you kind of in those initial stages when you were starting to get excited about science, and things that kept you from feeling discouraged about the career?
Yeah, I always feel discouraged. I always felt that it was impossible, but I also had some really amazing supporting parents that were always behind me and always supporting my work and telling me and repeating to me that I was capable of doing it. It's not because you have learning disability that you cannot achieve your dreams. You just have to work harder and also to learn how to work and try to avoid the issue by finding ways to work better using different tools, basically. So for me, it was focusing on molecules. And I guess for some other, it's finding a picture or reading loudly. So I think you have to find your motivation and the way to improve your learning abilities.
I think that's helpful to think about these tools and strategies that you can leverage to really move forward. And I think it sounds like this internship that you had in England was sort of a pivotal moment for you. Can you talk us through what brought you to that point, to that internship, and what was it about parasitology that really captured your interest?
So yeah, definitely this internship in the laboratory of Mike Donaroff in Bangor, North Wales, was really important for my life and my career. It was an Erasmus exchange program, so this university and scientific program exchange organized by the European community. And I met not only an amazing mentor that supported me, but also a lot of other young scientists like me coming from all over the world and giving me the opportunity to see the world and to open to the world as a whole. So not only as a French student, but as an international student, and also looking at other aspects of science and the fact that there is plenty of problem all over the world that can be solved if you think a bit outside the box. So it's not only cancer, it's not only neurodegenerative disease disorder, but there is also still plenty of disease in the world that is problematic for a lot of people. And then, yeah, these parasites are amazing. They change hosts, they are transmitted by either snail or mosquitoes or ticks. They change shape, they change form, and they can adapt to so many different conditions and hosts that I really was interested in and trying to understand how and why. How they were adapting and how they could survive and how we can actually find new treatment to fight this disease.
I think that's phenomenal. So it sounds like your heart was set at that point in pursuing a career and doing research in this area. What were the steps that you took then to ultimately get you to your current position?
So after this internship, which I highly advise young students to actually travel the world and see what's in the world and test different labs and different educational system, I really was motivated to really focus my career on parasitology. So apply for a parasitology graduate program, master program between the University of Lille and Oxford, and did a master. When you're in Europe, you first have to do a master, especially in France, before you can start your PhD. So I applied to different master programs, was accepted, of course, because I had already training. I was able to speak English by that time, and end up in a lab working on another type of parasite, a fungus infecting the lung of immunosuppressed patient called Pneumocystis carinii. And Pneumocystis carinii is the parasite, the fungus that helped scientists to discover HIV. And when I was working in this lab in the early to mid-90s, AIDS patient was still a huge problem, and pneumocystis and generating this large pneumonia in HIV patient was still a significant issue. So here again, I was excited to work on a pathogen that was still at that time killing a huge amount of patients infected with HIV.
Certainly. Well, it sounds like this was a particularly meaningful project and you were able to take these different steps. Then what ultimately brought you to Riverside? So you mentioned you did this master's program. What happened next?
The Pneumocystis carinii was an interesting parasite, but extremely complicated to work with. As of today, we still cannot grow the parasite in culture. So for someone really interested in molecular aspect of a parasite, it was difficult to work with this parasite. And with the advance of tree therapy, AIDS patients were surviving much longer, and pneumocystis was not as problematic as it used to be in the early 80s or early 90s. So I was extremely motivated to pursue my career in parasitology and decided to actually pick up the parasite that was still one of the most infecting agents in the world and decided to go for malaria and Plasmodium falciparum, which is one of the number one infectious disease killer in the world with tuberculosis and HIV. And as of today, I'm still working on the human malaria parasite. I did my PhD on it, so back in France, trying to understand cell cycle and cell cycle regulation, focusing on kinases and kinase inhibitor. Then I moved to the Scripps Research Institute and worked with Professor Elizabeth Winsler. There we developed microarray technology. It was a very exciting time in the history of science because we had early access to genomic information, whole genomic information, and started to do a bit more functional genomics and develop with the first microarray experiments to start to understand which transcript was expressed at different time of the parasite life cycle to find out how the parasite was controlling its development and progression into different hosts and different stage of the disease in human.
Oh, that's really interesting. And then I know next we mentioned in our introduction, you moved to the Genomics Institute of the Novartis Research Foundation. And this is maybe like a little bit different from your traditional academic postdoc experience. Can you describe what it was like working there?
Yeah, at that time, I just was unsure if I really wanted to stay in academia or if I wanted to work in industry. So working at the Genomics Institute of the Novartis Research Foundation was a perfect combination to see if I really wanted to pursue my career in industry and academia. And I worked officially there for 2 years, developed the malaria drug discovery program together with Elizabeth Winsler, and then decided that it was not for me. Decided that I really lacked trying to develop drug screening and setting up the screening for millions of molecules, but I was more interested in the how and why. I was more interested in a mechanistic understanding of how the parasite will survive inside a human body or mosquito's vector. So I decided very quickly that the industry was, while interesting, was definitely not for me because I had many unanswered questions that I wanted to answer myself, and decided quickly to actually apply for academia position. And that's how I moved to the University of California, Riverside between 2005 and 2006 to develop my own lab and to develop a lot of new technology to do a bit more advanced functional genomics and better understand the parasite life cycle progression.
Wonderful. Well, Karin, I know you are doing some really cool research in your lab today. So for listeners out there who aren't familiar with your work, can you share with us what is a project that you are working on right now that you are just the most excited about?
It's difficult to actually cite one project because I have more than one project that I'm very excited about. I have a project that are more focused on basic science, again, understanding the why and how, where we try to understand the mechanism of epigenetic and chromatin structure, so how the parasite regulates its gene regulation for its developmental progression. So this is for me really exciting because I develop a new tool, a new platform to understand how the parasites survive and which molecular components are essential for the parasite to survive inside its host. And there is another aspect of my research, and we recently published a paper in Science where we also still do a bit of drug screening, and I focus on natural product to identify new compound and new medicine against the human malaria parasite. So we start with natural product. I screen a lot of those. We also, I worked in collaboration with chemists where we try to simplify it a lot, this natural product, to make the compounds more efficient, but also easier to synthesize and cheaper, because that's the main issue. When you work with a tropical disease, when you work with disease that affect people living in developing countries, you have to make sure that not only you find an efficient new medicine, but this medicine is also cheap to produce and can be accessible to most living in this world. So we really do a lot of hard work at the organic chemistry level. And then when we have something that we think can do the work, my lab has recently developed a new functional genomic pipeline to take a systems biology approach to understand the drug mechanism of action of this new drug, new medicine, new compound, and figure out how this natural product worked and how this product is efficient at killing the human malaria parasite in human and not at killing its host.
I think that's very interesting. And I came across your science paper looking at this MED6189. Can you talk a little bit more about the findings and why this is so significant? Because I understand that not only does it work, but it also has a unique mechanism.
So yeah, it's derived from a natural product, which is always good. Still, the most efficient medicine against malaria are derived from natural product. This was one of the first molecules against malaria, quinine. We also have artemisinin that are also derived from a natural product. And the advantage of this product is the parasite take much longer to actually develop drug resistance against this compound. And the reason it takes much longer is because I think evolution has done the work. These natural products that are extracted from plants, from trees, or from marine sponges have done the work in trying to find the best way to kill pathogens infecting the plants or the sponge. And they have a more complex drug mechanism of action that the parasite take much longer to develop resistance to it. And this MED6189 is still a complex molecule to synthesize, still a bit expensive to produce, but has a unique drug mechanism of action targeting what we call the apicoplast. The apicoplast is a parasite-specific organelle. If you can compare, you have different organelles in a cell. You have the mitochondria in an animal cell, and in plants you have the chloroplast, very important to generate energy. But in apicomplex and parasite, including Plasmodium and malaria parasite, you have both mitochondria and apicoplast that are very important in the parasite to synthesize some important metabolite essential for lipidic pathway, vesicular trafficking. And without this apicoplast and with this parasite-specific organelle, the parasite cannot survive. And this new drug, derived natural product, MED6189, target this parasite-specific organelle. So it's definitely not toxic for the human host because this compound cannot target the human, but target something that's parasite-specific. And not only target these several proteins inside this organelle, but it also target the vesicular trafficking, so the molecular components that are essential for the biogenesis of this organelle. So a complex drug mechanism of action, but it's very, very difficult for the parasite to evolve resistance mechanism because it's targeting more than one protein and more than one metabolic pathway.
Well, Karine, I think this research is absolutely fascinating, and you can just start dreaming now, I think, of the potential impacts of this line of work that you're doing. But I think not every day in science is full of great discoveries and successes. I think, as we alluded to earlier, there are challenges, struggles, failures along the way that most people often don't get to see because only those final products are brought to light. So do you have a story of a challenge that you faced or a struggle or a big failure that you could tell us about today and kind of walk us through how you got through that tough time?
Yeah, I mean, success can only happen because of a repetition of a high number of failure and rejection. So you only see the good part of this project, but this was years and years of hard work. Years of rejection. When you actually write grant application to fund a project, very often you get rejected. So again, consistency, persistence, and then very often you take this rejection in a personal way, and you should not. You should realize that every successful scientist face many rejection, failure, and you have to repeat over and over again. I think extraordinary discovery really come from a high repetition of ordinary experiments that fail, and then you have to repeat over and over again and adapt your research a bit more each time and find a way to success. But yeah, hard work, persistence, and motivation.
Definitely. So perhaps to give us a sense of scale, do you remember how many years it took for this project to get to this point where you had a really exciting result to share?
It took more than 8 years.
Oh, wow.
8 to 10 years. And then often I have some of my colleagues who say, but how can you be so successful? And I will, well, before my first grant was funded and my first project was funded, it took me 70 grant application to finally be successful and to finally, so I had to go through 70 rejection.
Oh my goodness.
The funny thing is you don't get used to rejection. You actually don't get used to failure. The only thing that may change is you're usually mad and depressed for a day or two instead of being depressed for a week or a month. And then you quickly forget or try to forget and then go back to your lab, go back to your computer and start over again. I think if I have one advice to give to young scientists is don't be desperate and just keep trying again and again and again. And even if you get a rejection, even if you get no, just come back and be persistent.
Oh, I think that's such an important message. And I think important for people to realize that no matter what stage of your career you're at, you are going to get these rejections. And it might feel a little bit personal every time, but it's just part of the process.
Yeah, it's part of the process and it's also part of the success. I don't think there's any success without failure. And then when you finally manage to make it and you finally get a nice story, a nice paper and a result published, you're really happy. You're really happy for you, but also happy for your team, your collaborator. You have also to keep in mind that you're not alone. I mean, this is a large collaboration. There is no way you can actually do this work without a very strong collaborator doing the chemistry, the computational analysis. I mean, you're not alone. And the success is really a team success. It's not only you, but it's a combination of amazing colleagues, amazing scientists, and amazing students too. I have a really good student and postdoc that work with me on this project, and I have to be thankful to them too.
Definitely. And Karine, I love this idea of a team success and really celebrating the contributions of each person, because I think you're absolutely right. These successes have a long road involved and it's very difficult. There are a lot of challenges, but when you get there, I think you need to take a moment to celebrate, even if it's just a small victory. So we love celebrating these successes on the show. And I'm wondering, do you have a favorite success story from your career or your research, or even just a small but meaningful one that you've had recently that you'd like to tell us about?
Well, this recent paper published in Science really make me happy. because it's a nice story. Yes, it took a long time, but I was relieved and happy to show that even if you face difficulties in trying to identify the most complex drug mechanism of action or drug mechanism of resistance, if you use a combination of collaborator and team and essays, you can actually be successful. So the story does not end, which is always also good. We still have a lot of work to do. We still have to find a way to simplify the compound. But yeah, this slight moment of success was very, very important to me to demonstrate that, yes, my team and my collaborator at the University of California, Riverside can be successful in getting things done. And hopefully giving soon, we still have a long time to go, but giving a new possibility and hope that we can eventually find new treatments to fight against parasite resistance to all antimalarial. The way I usually celebrate is I like to invite my friends and have very good food and drink champagne. That's also one of the things that I like. I know I should not say that, but I like to drink a glass of champagne. When I finally reach a success, it's always a pleasure to share the success with your colleague and your family and friends that are always there during bad time and good time. It's not only good time. We had plenty of hard time to share too. So sharing good moments with your close one is also a very important moment of your life.
Oh, I think that is a huge win, and I'm glad you took the time to celebrate. And I'm also curious, I must say, you mentioned it took 70 grant applications to get a success. How did you celebrate that 71st application that was successful, or what was that moment like for you?
It was a big relief, and in a funny way, after this success, I also managed to finally get pregnant and give birth to my son. So it was a combination of a really good vibe and positive energy that also managed to give me the best things that ever happened to me, my son. So that was a very special moment.
Oh, that's wonderful. I appreciate you sharing some of these stories of your failures and your successes. I think it's a huge part of science. And I know some of these successes, as well as the failures, can motivate you to really dig in deeper in the lab and spend all your time at work. But we try to encourage our listeners out there to take a break, to read for fun, to sort of broaden their minds. So do you have a book that you've really enjoyed reading that you can recommend for all of us today, whether it's related to science or not, Karine?
I usually read, but I read French book. Saint-Exupéry is actually one of my favorite authors with The Little Prince, which I like a lot. And my son is named Antoine for Antoine de Saint-Exupéry.
Aww.
I also do a lot of yoga. I love yoga. I think this is also giving me some peace of mind and allow me to try to relax, remove the stress, and be able to actually take care of myself and take care of my brain when needed.
Oh, I love that. Well, we will definitely add this author recommendation to our website for our listeners to find there. And then one of the things we haven't really touched on, but is definitely a big part of your career, is travel. Even in talking about your career pathway, you've studied at all of these different places and now are, of course, in the United States from France originally. Can you talk a little bit about some of your travel experiences and perhaps if you have a favorite place that your science has taken you, whether it was for a conference or another purpose?
I think traveling and the fact that I decided to leave France is the reason why I've been successful. I think traveling open your mind and help you realizing that there's far more than just your country and your own little world. Help also you to accept other culture, other countries, other religion. Traveling is really important. So I will also advise younger students to take a break after your initial study or after your master to actually travel and see the world out there. It's inspiring and also help you to understand that you're really, really small in this amazing world and seeing beauty around you and different people open your mind and your heart. So traveling has always been an amazing aspect of my life. I like traveling to Africa. I like traveling to Asia. Bali is pretty. One of the best islands that I've been to is actually a very small island next to New Caledonia. It's called Pine Islands. I think it's one of the most beautiful and peaceful places in the world. I was there for many, many years ago, not for work, but for pleasure. But yeah, traveling is a very important aspect to anyone's success. And you have to stay humble and you have to realize that it's not only you. There is plenty of people around the world that can actually bring you so much more than what you think.
Oh, I think you're absolutely right. Travel can really help put things into perspective. And this Pine Island sounds secluded and wonderful. Do you have a favorite trip that you've been on for science, or a favorite place that you've been able to visit for science?
My favorite meeting is in Woods Hole, and a bit north of Boston. It's a molecular parasitology meeting. It's every year. It's in September. We talk a lot about different parasites: Plasmodium, Toxoplasma, Babesia, Trypanosome, Leishmania. And I usually meet all of my colleagues once a year, and we share our scientific discovery and struggle. I also like to mentor young faculty and let them know that success is not easy and you can do it. So there's a lot of things that I like to do outside my lab, and meeting your colleague and meeting especially young faculty and young women, and supporting this young generation is also something that I like to do a lot at the moment.
Oh, absolutely. And I've heard Woods Hole is an absolutely magical place to be talking about science and surrounded by all these people in the field who share your interests. What is it like there? What was it like when you were there last?
I was there last just in September, just a few weeks ago, and I didn't present any data. I actually like this meeting because it's mostly student and postdoc presenting, and you see the new generation raising, and it's just giving you hope for the future. And again, supporting younger generation and supporting women in science. As a woman in science, I faced some additional issue when I was younger, and I also want to do what I can to avoid this challenge for most of the younger women and younger generation. So giving them support, giving them hope, and give them the key to success. So do not get too depressed by rejection and failure. And again, persistence and hard work will always be the key to success.
Definitely. And I'm so glad to hear that you're valuing these mentorship experiences and being able to go out there and support the next generation of scientists, particularly women in science who are facing a lot of challenges. And I think the people who you meet in science are absolutely remarkable, whether it's at these small meetings or even in your own department at your university. So I think there are stereotypes out there, Karine, about what scientists are like that you see in the media, on movies and TV. But I think so often they're not true. A lot of the people that I meet in science are funny, creative, amazing, friendly people. And that's not often how we're portrayed. So do you have a story or an example that really shows this human side of science, whether it's just funny or fond memories or a fun tradition that you had in a lab, something like that, that kind of goes against the stereotypes that people may have?
I know, especially at the moment, science and scientists are not always valued the way they should, but scientists are not all of them, but often very humble and not always indeed boring, but I've been often described as a geek, but I think we are in general simple and really care about the truth. And trying to understand how the world works often doesn't link to a high-paid job either, but how that it worked and how we can work together and collaborate is, I will say, collaboration and discussion, being open and being humble sometimes. Humble when we actually look at the scientific problem and take years to answer a specific question. So when you're trying to solve a problem, you realize how small you are sometimes. But I really like to meet with my student, my colleague, and support them. And yes, we usually get also good food and share drinks and a nice story, and supporting each other is really something that I appreciate. Again, it's not for everyone. There's always some people that are more challenging to deal with than others. But in general, I will say, and I have to say, I think the young generation seems to be also more open and more inclusive, I will say. So I'm happy about that.
Absolutely. And I think this collegial, collaborative, friendly vibe that you're depicting here is against the stereotypes. A lot of people think about scientists as these cold, very serious, always working sort of people. So I'm really glad to hear that you've developed this warm and welcoming network of people that you're able to kind of bounce ideas off and work hard towards, like you said, finding the truth.
Yeah, and collaboration. I really think that in the past, and still when I arrived as a young assistant professor, people told me, you have to write your own grants alone to show your independence. And the type of work that I do are large functional genomic type of study, and you need collaborator, you need different type of expertise. And you cannot work on this large project alone. So collaboration between chemists, computational biologists, MDs, and molecular biologists are essential in our world today. And I think the time where everyone was working alone in his own lab is long gone. And I do value collaboration. I know there is competition, and I have a hard time with competition. I'm not great at that. I rather work with people than compete against people. So collaboration for me is also key to success.
Definitely. I think having the right collaborators can really help you take those important big steps forward in your research. And I think oftentimes you are still limited, however, by different barriers, whether it's funding, staff, technology, feasibility, or even hours in the day. So, Karine, if we could give you everything you could dream up, is there one particular research question or problem that you would want to address first?
I still didn't answer my question on how exactly the malaria parasite managed to survive inside the human red blood cell and all of the molecular components that regulate cell development and progression. So I'm still working on it and try to do a bit more comparative genomic analysis to analyze what's specific to the parasite compared to other apicomplexan parasites. Of course, we always need money, but time, it's going too fast. So getting more time and still more support from administration will be amazing.
Oh, absolutely. I think just having unlimited hours in the day, you could accomplish so much. And I'm really glad to hear that you're able to work on one of these questions that you are just so passionate about. And I think that is a key driver for a lot of people who are working in science today. It's just they're so excited and curious about the work that they're doing. So for our listeners out there who might be on their own scientific journeys or maybe just at a fork in the road in life, do you have a piece of advice that you could share with them that perhaps really helped you on your own journey, Karine?
Be proactive. Go and talk to people. Meet with scientists and ask them to do an internship. Ask them to mentor you. So go out in the world and leave your phone behind and leave your connected world behind and go and start and talk to people and open your eyes. And don't be afraid of failure.
Well, I love that advice. I particularly like the leave your phone behind. I think it's so easy to feel glued to that, watching these emails pop up and just multiply in your inbox, but just really disconnecting and being in the moment and connecting with other people around you, I think, is something none of us do enough.
I love talking to people. I think when you talk to people, you come up with new idea, new solution, a new problem also. That's why I love going to meetings and learning from other people, because if you stay on your own, if you stay in front of your screen, and if you stay in your office alone, I don't think you're going to make huge progress today. And especially with AI, it's very often scary what's going to happen in the next few years. But I think connection, talking to people, becoming excited, and traveling and looking at the world problem today in the eyes of others is really important. Similar to, you have to talk to people in your lab, you have to talk to your collaborator, but it's also very important at university to talk to the staff. I think that's my major issue today. We lose this connection, we lose this social interaction. I mean, I can only be happy if I am actually meeting and talking to people. And I think this has also been a key to my success. Going forward, meeting people, talking to people, and eventually thinking a bit more to myself and getting this feedback and getting new ideas.
Well, I think that is a wonderful message for listeners. Karine, is there any other piece of advice you'd like to share, or perhaps a last note of inspiration that you would like to end our conversation on today?
Science is great, but getting out in the world is also very important, and integrate also your family and loved one in your success story. They are very important. And so work is important, for sure, very important for me, but social aspect of my life and taking good care of myself is also very important.
I love it. A perfect note to end on. And if our listeners would like to learn more about you and more about the work that you're doing, Karine, what is the best way for them to do that?
So they can actually go to my UCA webpage and my LinkedIn account, where I often post some of my most recent research articles. And if they are interested in talking with me, especially for this young woman in science, please send me an email. We'll be happy to answer your question, to mentor you if you need some mentorship. Always happy to actually support young scientists and young women in science from every place in the world. And just reach out to me and I will be happy to help in the best way I can.
Well, much appreciated, Karine. Listeners, definitely don't hesitate to reach out and get in touch and learn more about the amazing work that Karine is doing in her lab. And Karine, it's been such a pleasure to chat with you today. Thank you so much for your time.
Thank you so very much for your time as well, and hope we can talk again soon.
Oh, absolutely. I enjoyed our conversation, and listeners, always wonderful to have you here with us as well. We'll see you next time on another episode of People Behind the Science.